Good morning all. Today we will discuss about the phases of the clinical trial, what is clinical trial, and there are some rules which ultimately governs the clinical trial. So ultimately let's just start with the clinical trial. What does this clinical trial means and why this clinical trial this is needed for us? First, you know a famous logician is Kurt Gödel. Kurt Gödel famously said once that I don't believe in empirical truth, I only believed in priori truth. That is particularly more important in case of the drug, in case of the medicines, particularly when it is used in us, used in humans to treat or to diagnose different types of the diseases. That is, we need some a priori truth, that is, we need some evidences, we need some guidelines that ultimately dictate these evidences that these medicines are ultimately safe as well as the effective in case of the humans. So ultimately all these can be done by means of the clinical trial. So there are thousands of the compounds, thousands of the chemical compounds, for example, for which by different types of the mechanisms we can choose some molecules that can act on some molecules in our body to treat or to diagnose some diseases. And from these compounds are ultimately made to use in different types of the preclinical studies or preclinical trials. In preclinical studies, these drugs are given to animals, different types of the animals, and accordingly their pharmacokinetics in those animals are ultimately inspected. The pharmacodynamics, that is how it is used in those animals, are inspected, and ultimately from this a dosage that can be used in humans can be calculated from these preclinical studies. And after the preclinical studies, if the drug is fit to use in case of the humans, then comes the clinical trial. So clinical trial is done on human subjects ultimately to inspect its efficacy as well as the safety. And there are different set guidelines known as the good clinical practice that is mainly needed to ultimately safeguard the rights and safety of the trial subjects, that is those participants or subjects on which these clinical trials are done, and also the clinical trial data are credible, they are valid, they are credible, so that can be used and these drugs can be marketed, so that can be used, widespreadly used in different types of the individuals to treat disease or to diagnose some disease, etc., etc. So this is the basics of the clinical research or clinical trial and why the different guidelines are needed in case of the clinical trial. So you can see this is the definition of the clinical trial. So any investigation in human subjects, so clinical trial is done in human subjects. Before that, I have already said that this is some studies are done in preclinical studies in case of the animals. So first the compounds are tested in animals to see its safety, its efficacy, its toxicity. There are different types of the toxicity studies are done on case of the animals, like carcinogenicity study, embryonic toxicity study, reproductive toxicity study, acute toxicity study, chronic toxicity study. These are done, and then if it is feasible, then it can be come to the human trials. So human subjects intended to discover or verify the clinical, pharmacological and or other pharmacodynamic effects of an investigational products. So pharmacological effects, clinical effects as well as the pharmacodynamic effects of an investigational product. This investigational product is a drug. So when the drug is not approved by our regulatory agencies, no clinical trial have been done, it is known as the investigational product or IP, investigational product, okay. And or to identify any adverse reactions to an investigational products and or to study the different pharmacokinetic parameters like absorption, distribution, metabolism and elimination or excretion of an investigational products. What is the object? With the object of ascertaining its safety and or efficacy. Both the safety as well as the efficacy, these two are the important when a drug have to come to the market. The terms clinical trial and clinical study are usually synonymous. So clear, clinical trial means when a new drug is known as the investigational product. That investigational product, that may be a drug, that may be a device also. You know that there are different medical devices also. When these are tested in case of the humans, why it is tested? To see whether if there is any toxicity, if there is any changes, what is the changes in the pharmacodynamic as well as the pharmacokinetic parameters. Pharmacokinetic parameters mean when this new drug or investigational product is given to humans, how it is absorbed, how it is distributed, how it is metabolized, and how it is eliminated from the body. And pharmacodynamic means how it acts on different receptors, or it acts on different enzymes, or in different second messenger system to ultimately do some effects, and these effects are needed to treat the disease. Okay, so that's why the clinical trial is needed. So you can see basic research is ultimately dictates the answer of how does nature work and what causes the disease, and from this basic research we can identify that these are the diseases, and this disease caused due to the deficiency of this or due to the increment of this, so that we can do some intervention to treat this disease. So this is known as the translational research. How can biological knowledge be used to treat the disease? And ultimately this clinical research that encompasses the clinical trial is needed. Are these treatment strategies are safe and effective in humans? Both is important, because a drug may be very much effective for the treatment of the disease, but it is not safe. In 7 out of the 10 individuals, it will do some toxicity. So this drug should not be used like this. Again, some drug may be very much safe, but it is not effective. So both of these are important, safety and efficacy. And for this, the clinical trial or clinical research studies are needed. Now coming to the what is a new drug, investigational product, what we are talking about, a drug including active pharmaceutical ingredient or phytopharmaceutical drug, like if it is obtained from some plant products which has not been used in the country to any significant extent. That is, it is not used in our country, it is not used. It may be used outside, okay, except in accordance with the provisions of the Act and rules, etc., etc., and it is not approved as safe and efficacious by our central licensing authority. The central licensing authority is the DCGI, under which the CDSCO acts. So ultimately this DCGI and CDSCO grant permission for the marketing of newer drugs. So new drugs, if it is not present in our country, no clinical trial have not been done for that disease in our country. Or a drug which is already approved by the central licensing authority for certain claims and proposed to be marketed with modified or new claims including indication, route of administration, dosage or dosage form. What does it mean? For example, drug A is indicated for treatment of one disease. Now it has been some trial has been done in some outside countries, and it has been seen that this drug A can also be indicated for some other diseases also. So again, this drug, although it is approved in our country, again clinical trial has to be done for that indication to grant permission for marketing for that indication of the disease. For example, drug A is indicated disease A. Now for disease B, I want to market it. So again a clinical trial have to be done. Similarly, route of administration. For example, one drug can be used as a oral form. Now a drug is ultimately packaged in an injectable form. Again, we have to do some clinical trial, or we have to do some clinical research studies to see whether the injectable form is also safe and efficacious in our country or not. Dosage form, similarly, route of administration, dosage or dosage forms. So it may happen that this drug is approved from one indication, and for some other indication or for some other dosage form, we have to do some clinical research studies. Again, a fixed dose drug combination. Drug A, drug B and drug C. These three drugs are individually approved in our country for marketing for some diseases. Now, if we want to combine these three drugs together in a single pill, for example, this is known as the fixed dose drug combinations. Already we have seen in the general pharmacology class. So again, we have to do some clinical research studies to see whether if these 2 or 3 drugs are combined together, whether is there any problems in the drug-drug interactions, whether is there any effect of this combination of the drugs on the pharmacokinetics as well as the pharmacodynamics. Okay, that's why again some clinical research studies have to be done. Modify the sustained release form of a drug or novel drug delivery system. For example, a drug is present in a oral drug or injectable drug. Now we want to use as a transdermal patch. So it is a novel drug delivery system, transdermal patch. So again we have to do some research studies. Or some sustained release form. Sustained release form means we have to change the ultimately excipients, its packaging, so that there will be sustained release of the drug over a long period of time. Again we have to do some pharmacokinetic and pharmacodynamic studies to see whether it is safe and effective. It ultimately also encompasses a vaccine or recombinant deoxyribonucleic acid, that is the rDNA derived product, living modified organism, different types of the biological agents like monoclonal antibodies, biosimilars, stem cell derived product, gene therapeutic product, xenografts, all this comes under this drug, new drug, and we have to do the clinical research studies. That is not only the single oral pill. It may be some medical device, it may be some biological agents like monoclonal antibody, it may be some vaccines, it may be some graft. So all these have to be done, the clinical research studies for its approval. So you can see from this graph how does the drug development process happens and how much time it needed. Typically when a compound is selected and ultimately it comes to the market, it usually takes a long period of time, 10 to 15 years. So today I am just inventing or discovering some compound, that this compound may be druggable. Druggability of a compound, again this drug will come to the market approximately 10 to 15 years after, because it needs extensive clinical and preclinical research before it comes to the market, before it granted approval in different countries. Okay, so you can see first thousands of compounds are screened to see whether what is the druggability of a compound. Then the preclinical pharmacology studies are done, then preclinical safety studies are done, then clinical pharmacology and safety, that is the clinical trials are done. You can see this is the different phases of the clinical trial, and it approximately takes 10 to 15 years ultimately when it comes to the market. So thousands of the compounds are screened and ultimately only 1 or 2 products ultimately came to the market. So billions and billions dollars are needed for a clinical trial, because many drugs ultimately failed in the different phases of the clinical trial. The failure may be due to the toxicity, the failure may be due to the druggability, failure due to the efficacy. So thousands are screened and ultimately it comes only 1 or 2 products in the market. Okay. So these are the phases of the clinical trial, very important. Phase 1, phase 2, phase 3 and phase 4. There is another phase that is newer entrant, is the phase 0. So first I am starting with the phase 0. What is phase 0? Already I have said that when a drug have to come to the market, first we have to do some preclinical trials, that is we have to test in the animals. So after the animals, it has to be tested in the humans. So it has been seen that if we are starting with the humans with a large doses, that takes more and more money, and ultimately if it fails, so ultimately failure will happen. That's why phase 0 means it is a micro dosing study, that is very minute subtherapeutic quantity of the drug is given in human volunteers to see whether there is any adverse effect or not, to see some preliminary ideas about the pharmacokinetics as well as the pharmacodynamics. This is known as the phase 0 studies or the micro dosing studies. This is usually done nowadays to ultimately cut off that if the phase 0 studies you are seeing that this drug may not be beneficial in the long run, we can cut off this from the further progress. Next, coming to the phase 1 study. Usually most of the compounds, we usually after the preclinical studies, we directly go to the phase 1 study. So phase 1 study is the human pharmacology study. What happens? In phase 1 studies, the drug, the new drug, investigational product we usually tested in healthy volunteers, not in the diseased individuals, except in few, except in very toxic drug, that is those drugs which are used in oncotherapeutics, that is anti-cancer drugs. Since these are the very toxic, only these drugs are used in the phase 1 level in case of the diseased individual. Otherwise, healthy volunteers, healthy individuals, we are testing the drug. Why it is testing? Mainly to see the safety of the drug as well as to ascertain the dosage of the drug. So we are giving the dose that is calculated from the animal studies, that is this drug dose can be beneficial in case of the humans. That is also calculated if we do some phase 0 studies, microdosing studies. Now this dose is given in healthy volunteers, and for particular interval of the time we are taking their blood, we are taking their urine, we are taking their stool sample, etc. to see what is the concentration of the drug. We ultimately plotting the concentration time curve, what is the bioavailability of the drug, what is the volume of distribution of the drug, tissue distribution of the drug, protein binding of the drug, metabolism, how it can be metabolized, and then how it is eliminated, whether it is following the first order kinetics or whether it is following the zero order kinetics. So all this information in case of the healthy volunteers can be available when we are doing the phase 1 study in case of the in healthy volunteers in clinical trial. And also we are seeing if there is any toxicity in those persons or not. We are giving this dose, and if there is any toxicity is happening. So first human administration of the drug is carried out, it is carried out by qualified clinical pharmacologists or trained physicians in a controlled setting. So it is done in a controlled setting where all vital functions are monitored. We are also monitoring their blood pressure, we are monitoring their pulse, respiratory rate, temperature, etc., etc. And if there is any emergency or if there is any problems, there should be adequate resuscitative facility should be there where the phase 1 clinical trial is going on in case of the healthy volunteers. Subjects are mostly healthy volunteers. They are exposed to the drug one by one. Usually the total number of participants ranges from the 20 to 80. They are starting with the lowest estimated dose, and if there is no toxicity, we are gradually building up the dose, increasing the dose to see what is the maximum tolerated dose. Okay. So ultimately stepwise to see the maximum tolerated dose and effective dose. The emphasis is on safety, tolerability. So safety, tolerability is the main thing that we want to see in case of the phase 1 clinical trial, and to detect any potentially dangerous effects on vital functions. That is, if this drug is given in some doses in healthy volunteers, that increases the temperature or that increases the respiratory rate. So we can note this like this. The human pharmacokinetic parameters of the drug are measured for the first time in phase 1, or if there is phase 0, there may be microdosing. Also the human pharmacokinetic data are measured, but remember in phase 0 trials, sub-therapeutic concentration of the drug is given. That is very minute quantity, like 10 microgram of the drug is given, where the original dose that is needed for the treatment of disease approximately 10 milligram. So approximately nearer to the therapeutic concentration drug is given only in case of the phase 1. Now this phase 1 we are giving the drug and we are testing what is the pharmacokinetics of the drug and also what is the safety and tolerability of the drug. Okay. So the human pharmacokinetic parameters of the drug are measured for the first time. Usually no blinding is done. That is, the investigator knows that this drug is given. Usually it is a warm-up study. The drug is given to the healthy volunteers and tested. Their different samples are taken, pharmacokinetic data, toxicity data, tolerability data are ultimately tested. The study is open label, that is everyone knows that this drug is given in phase 1 study. So the main important thing about the phase 1 study you have to remember, very important, phase 1 study in most of the cases it is the first in human study if the phase 0 study is not done. Second thing is phase 1 study mostly done in case of the healthy volunteers, not in case of the diseased person. It is done in case of the diseased person only if the drug is very much toxic, That is usually done in case of the oncotherapeutics, that is in case of the anti-cancer drugs. Third is phase 1 trial is mainly done to see the toxicity, safety, tolerability as well as the pharmacokinetic data in case of the healthy individuals. Okay. Now the data are gathered. It is seen that the maximum tolerated dose, effective dose, we are ultimately ascertained from the phase 1 study, and it is seen that this drug may be tolerated or may be safe in case of the individuals. Now we proceed to the phase 2 trial. So phase 2 trial is also known as the therapeutic exploration and dose ranging studies. So phase 2 trial, in phase 2 trial, first we are testing the drug in case of the diseased individual. Now for which disease this drug is indicated, we are giving this drug. Okay. For example, we are giving some drugs for some purpose. Previously in phase 1 study it is given in case of the healthy volunteers. In phase 2 studies, first it is given in some diseased individual. Okay. So this is conducted by physicians who are trained as clinical investigators. It involves 100 to 500 participants selected according to the specific inclusion and exclusion criteria. So there are inclusion criteria, there are exclusion criteria. So ultimately, according to the inclusion criteria and exclusion criteria, some participants, some diseased individuals are chosen who are voluntarily enrolled themselves into the clinical research studies or clinical trials. Okay. The primary aim is the establishment of the therapeutic efficacy. Now first we want to see the therapeutic efficacy because it is done in case of some diseased individual. Previously we have done this in healthy individuals, so there is no question of efficacy. Now we are seeing whether the disease can be cured or not. First efficacy studies can be done, as well as the dose ranging study. We are also testing the different doses, the lower dose as well as the higher dose, whether there is any change in the efficacy as well as the safety or not in a control setting. Tolerability and pharmacokinetic data are studies as extension of the phase 1. Now we are studying also tolerability and also studying the pharmacokinetic data in case of the diseased individual, and we are comparing, that is, if there is any change in the pharmacokinetics from the healthy ones to the diseased ones or not. Because when there is disease, for example some liver disease or some kidney disease, there will be ultimately change in the distribution of the drug or absorption of the drug or metabolism of the drug or excretion of the drug. So ultimately that also dictates the dose that is needed in case of the diseased individual. Okay. So pharmacokinetic data are tested, tolerability data are tested, and in addition in phase 2 usually we are seeing the efficacy data as well as the dose ranging data. The study is mainly controlled and randomized. Randomized means the allocation we do not know, the investigator do not know in which arm the drug is given. In the other arm usually the placebo is given in phase 2 trial, or some standard drug can be given. So there may be some for example 10 participants are enrolled in phase 2 clinical trial, 5 participants are given the newer drug, 5 participants are given some placebo drug. Placebo means that some drug is given but they do not have some pharmacodynamic effects, or some standard drug is given. Now this allocation between these 5 and 5 is random. That is, the investigator do not know which participant is getting which type of drug. This randomization is needed because often the investigator may be biased, because the patient came with some severe disease, which may be biased that we do not know about the efficacy of the newer drug. We should give some the standard drug in this patient. So this is known as the biasness. So if we are biased, ultimately the results will not be valid, the results will not be precise. So we need randomization. This is known as the randomization. That is, we do not know in which patient which drug can be given. So this is known as the randomized. And maybe blinded and open label. Blinded means the investigator or those who are analyzing, they also do not know which drug is given to the which participant. Participant do not know about, they are taking, because I am a participant, I am taking a white pill for as a trial participant. I do not know whether this pill contains the newer drug or this pill contains already a standard drug or some placebo, that is no drug at all. If the participant do not know about this allocation, this is known as the single blinded study. That is, the participant do not know, but the investigator, that is the doctor or the analyzer, know. When the investigator, that is the doctor, I am giving this two pills to two patients. Both are similar looking. I do not know which pill contains the active drug, which pills contains the placebo drug or already standard drug. The participant also do not know which is the standard drug, which is the new drug. So both I as well as the patient do not know about the allocation. This is known as the double blind study. That is, both of us are blinded about the allocation of this drug. There is another is the triple blind. That means those who is ultimately analyzing the whole the data, that is the statistician or the analyzer, they also don't know about which person got the which one. They only know about the code. That is, this person got drug A, this person got drug B, this person got drug C, etc. This is known as the triple blinded study. So these are mainly done to increase the statistical appropriateness. So statistically we need a strong evidence for the approval of the drug. That is, this drug is safe, this drug is efficacious. This needs the statistical analysis, and for this statistical analysis to be unbiased, we need this type of the blinding, this type of the allocation concealment, this type of the randomization, etc., etc. So it is generally carried out at 2 to 4 centers. The candidate drug may get dropped at this stage if the desired level of the clinical efficacy is not obtained. Most of the drugs that ultimately came to the clinical trial stage fails in the phase 2 trial. Okay, so only fewer drugs proceed to the next phase, phase 3. So after the phase 2, next coming to the phase 3, which is the therapeutic confirmation or the comparison study. So phase 3 is the therapeutic confirmatory trial that is done in a multicentric way, that is different centers. Usually it is done in different areas. If it is a multi-country studies, it is done in different countries. If it is done in our country, it is done in different states to ultimately include different types of the ethnicities. This also contains different types of the inclusion criteria and exclusion criteria. That means this age group should be included, this type of the disease should be excluded. That is, we only want to test this type of drug who do not have liver disease, like this. So this is known as the inclusion criteria, exclusion criteria. Multi-center studies, large number of the diseased individual, approximately 1000 to 3000 or 5000 participants are enrolled within this study. This is mostly again the randomized study, control studies. Usually there may be 2 arms. In the one arm, the newer drug is given, the investigational drug is given, and the other arm, already the drug that is already approved or the standard treatment guideline, it is already present, it is given. If there is no standard drug, we can give some placebo. Again, placebo means that does not have that type of the pharmacodynamic study. So it is a double blind comparative trials conducted on a larger patient population by several physicians at many centers, several centers. Safety and tolerability are assessed on a wider scale, while pharmacokinetic studies may be conducted on some of the participants to enlarge the population base of the pharmacokinetic data. That is, the pharmacokinetic studies, pharmacokinetic data are continuously taken, safety data are continuously taken. Indications are finalized. So ultimately, this is a confirmatory, therapeutic confirmatory trial. Indications are finalized, guidelines for the therapeutic use are formulated, and after this trial, a new drug application is submitted to the licensing authority, giving all the data of the previous ones. That is, we are giving the data of the preclinical studies, we are giving the data of the phase 0 or phase 1 studies, phase 2 studies, and lastly, everything about the phase 3 trials. This is known as the new drug application. Then this is submitted to our licensing authority, DCGI. Okay. So ultimately, if they are convinced, then the marketing permission is given. Marketing permission means this drug can be marketed for this indication in this dosage form. Okay. So this is indicated, that can be marketed, and the patient can buy, the doctor can use this for the treatment of that disease. Next, coming to the phase 4 or post-marketing surveillance and post-marketing studies. Now the drug have been marketed. So after the marketing of the drugs, usually the licensing authority in all the countries needs some data after the marketing for approximately 2 years to 3 years. Because when a drug comes to the market, large number of the patient will take that, and the clinical trial is usually done in a controlled setting. That is, we are continuously monitoring the patients. We are continuously taking their samples, we are continuously taking their vitals, we are supervising whether they are taking the medication or not, we are supervising about their foods, we are supervising about their drugs. These are the inclusion criteria, these are the exclusion criteria, etc., etc. But when a drug comes to the market, when a doctor prescribes it, the patient can take, there are different types of the dietary habits in different patients. The patient can take different other drugs for different other indications. So there may be different types of the drug-drug interactions. There may be different types of the drug-food interactions. Since large number of the patients are taking, there may be more chances of some rare adverse effect coming out that cannot be captured in a controlled clinical trial for a short period of time. Again, if it is taken for a long period of time, some chronic adverse effects are also coming out. Okay, because we have done the clinical trial for a short period of time in a controlled setting. So newer adverse drug reactions, if it is present, has to be reported. So this comes under the post-marketing surveillance studies. So newer adverse drug reactions, newer pharmacokinetic data, newer any data have to be submitted to the licensing authority for approximately 2 to 3 years by the company who are marketing this. This is known as the post-marketing surveillance. Apart from this, there are other studies are also going on after the marketing. These are known as the other post-marketing trials. For example, after the marketing, the company or some academician purpose also, some academicians also, we are comparing this drug with some other drug. Because these two drugs are already approved. Some drugs already approved, this drug is already approved. We are comparing this. We can do some genomic studies or pharmacogenetic studies. Again, already we have seen about the pharmacoeconomics. So we are doing some pharmacoeconomic studies to see that whether this drug is cost effective or cost beneficial in comparison to another drug or not, or another procedure or not. We can do different other pharmacoepidemiological studies. So all these different types of the studies and trials can be done after the marketing of the drugs. This all comes under the phase 4 trial or post-marketing trial. So the difference is clear. One is post-marketing surveillance, that is regulatory mandated for approximately 2 to 3 years by our regulatory agencies, to ultimately the company who are marketing the drugs have to submit different data like different newer adverse effect, rare adverse effect, chronic adverse effect, newer pharmacokinetic data, etc., to the licensing authority for 2 to 3 years after marketing of a new drug. Apart from this, any other studies, any other trials, any pharmacoepidemiological studies, there may be pharmacogenomic studies, pharmacoeconomic studies, any other comparative studies, all these trials comes under the phase 4 trials, post-marketing studies. So uncommon or idiosyncratic adverse effects or those that occur only after long-term use or unsuspected drug interactions are detected at this stage. So this is also known as the effectiveness studies. So clinical trials is done in a controlled setting. These are known as the efficacy studies. Efficacy is when we are doing some studies in a controlled settings, supervised settings. And effectiveness studies means when it is done in an open setting. That is, it is present in the open market. Now we are seeing their effectiveness. So effectiveness we want to see in a open studies in case of the phase 4 trials. Patterns of the drug utilization and additional indications may emerge from the surveillance data. So from this data, we can see that the company also can see that these drugs may be beneficial in some other indications also. Because these patients have some other disease, they are also cured of this like this. So the company may think of starting some clinical trial for that indication of that drug. So this is the way by which the newer indications are added. Okay. So further therapeutic trials are also done in some other populations also, because this clinical trials is again done most cases in case of normal adult person 18 to 65 years. But when it comes to the market, it will be also used in adolescents, it can also be used in elderly persons, it is also used in different persons with different other diseases also. So here these small studies are also comes under the phase 4 studies. That is whether it is useful in case of the children or in case of the elderly, or if it is can be used in the pregnant or lactating women, patients with renal and hepatic disease, whether it can be given or not, etc. That may be undertaken at this stage. That is phase 4 studies includes all these things. So after the marketing of the drugs. Okay. So now coming to the good clinical practice. So all these things what we have discussed, this clinical trial, this has to be undergone by a set rules or regulations. These set rules or regulations are known as the good clinical practice. What is the definition of the good clinical practice? This is basically a standard for the design, conduct, performance, monitoring, auditing, recording, analysis and reporting of the clinical trials. That is everything about the clinical trials. How this clinical trial have to be conducted, how it is to be measured, how it is to be analyzed, how it is monitored, everything have to be done in accordance with this good clinical practice guidelines. So ultimately why it is needed? Because it provides assurance that the data and the reported results are credible. That is, credibility is there and accurate, and that the rights, integrity and confidentiality of the trial subjects are protected. This is very important. This is not such that we may include any patients জোর করে কাউকে ধরে নিয়ে ক্লিনিক্যাল ট্রায়ালে ঢুকিয়ে দেবো. This is not like that. They are not guinea pigs. So humans are not guinea pigs. What is done in case of the Nazi period? I am coming to this slide. So every trial participants have to be known about their disease. They have to give consent. This is known as the informed consent. This is very important and part and parcel of the good clinical practice. Informed consent means the patient have to be understood about the mode of the clinical trial. That is, they have the disease, this is the clinical trial, we are giving these newer drugs, or you can get the newer drugs or the already approved drug or some placebo, because it is blinded. Then we are testing these and these, we are testing these bloods, this follow up is needed, and if there is any adverse effect happens, we will treat this like this. Everything the patient have to be understood. Then this patient gives voluntarily consent that I want to be associated, I want to be incorporated in the clinical trial. This is known as the informed consent. That is, information is percolated to the patient. The patient have to comprehend about the information. It is not like that you are telling in English about the about everything about clinical trial and the patient does not know about anything, any bit about the English. So the patient cannot comprehend, the patient is ultimately made to sign. So this is not that. The patient have to be comprehended about everything about the clinical trials, then the patient gives consent. This is known as the informed consent. And the next thing about the participating in the clinical trial is the voluntariness. The patient have to be voluntarily included, and the patient also voluntarily exclude themselves at any point of the clinical trial if he or she deemed it feasible. So any point of time the patient can exclude themselves from the clinical trial. Okay, so this is known as the voluntariness as well as the informed consent. These two are very important in case of the good clinical practice. So why we need guidelines, I am not going to the details, but if we can see that there are different types of the irregularities in the clinical trials all over the world, in our country also, that ultimately leads to different types of the clinical trials. These are the newspaper cuttings that ultimately leads to problems. Ultimately deaths can also happen in the clinical trial subjects. That's why we need the guidelines, so that the right safety as well as the integrity of the study subjects will be protected. So right safety, well-being of the trial subjects are protected, and clinical trials that are credible. This is also needed. So we are doing some studies and the clinical trials that are not credible. Again, that drug comes to the market, and if it goes to the market, thousands of the patients, there may be some adverse effect or they may die. So the clinical trial have to be, again, it should be valid, it should be precise, it should be accurate. That is also needed. So the GCP guidelines, so Hippocrates first says that we should not harm individuals. So we are treating a patients for the benefit of the patients. Okay, that is already needed. So these are the different ways, there are different stages by which this good clinical practice ultimately comes. Details is not required, I am just giving a nutshell. That is, in 1906 first the Pure Food and Drug Act comes. So any regulation, whether it is needed, that first comes in the United States of America in long back in the 1906, and after that there is famous sulfanilamide disaster which occurs in the 1937. So basically sulfanilamide, it is safely used in tablet and powder form, but when it is used in a liquid form, that liquid is usually diethylene glycol, and it is mainly used to increase the appetite of the persons. It is marketed as elixir of the sulfanilamide, but due to the presence of this diethylene glycol, more than 100 people ultimately dies when this is used. So this is known as the sulfanilamide disaster. So these are the different incidents, or you can say accidents, ultimately that gives rise to the concept that we need some regulations. Okay. So in 1938 from the act that is more comprehended, that is becomes more precise, and Food, Drug and Cosmetic Act was ultimately formed. And you know about the during the World War II times, there are famous Nazi experiments. There are numerous Nazi experiments is done where the ultimately those persons who are in the concentration camp, they are ultimately different types of the medical experimentation was done on them without taking into account of their voluntariness or their consent. There is nothing. So German physicians, 23 Nazi doctors conducted medical experiments on thousands of the concentration camp without their consent. So consent is not needed, it is given, some drug is given. Most of the participants died or permanently crippled. So you can see, an American military tribunal, ultimately criminal proceedings was done later. 23 doctors were ultimately prosecuted. They had said that they had done some grievous crimes. So as a result, some Nuremberg Code was enacted. So the main part of this Nuremberg Code is what I have already said, that is the voluntary consent. That is, I want to be associated. You are not there to ultimately force me to be associated with a clinical trial. So Mengeles Tyndall, famous this one, the twins of Auschwitz. There are various twins and they are doing different types of the research to the twins, so that one of the twins or both of the twins ultimately dies. So Joseph Mengele is a famous doctor. He was known as the angel of death. Okay. Mengel's secrets of the hereditary, because they know what is the heredity of the Aryan race in case of the Nazis. So in the twins, because both of them have similar type of the genetic makeup, he is testing, he is giving different types of the drugs, both of to them, to see whether there is any changes or not. So ideas of the, so this is mainly done in case of the Auschwitz. So large number of the available twins during World War II in 1944-45, and many of them dies. Hence, he is known as the Joseph Mengele, angel of death. So trials, Nazi doctors are prosecuted for performing inhuman experiments. They have argued that there was no law suggesting these experiments should not be carried out. So what are their arguments? That there is no law. That's why, for the sake of our medical science, for the progress of our medical science, we are doing these studies, because they are prisoners. We are giving some drugs, we are giving something, because they are also percolated different types of the pathogens to the individuals. They are percolating syphilis or some pathogens, and they are giving different types of the injections to see whether they are healing or not, or whether they are dying or not, like this, simple. So not guilty at Nuremberg at first, they are ultimately, they are arguing, the German defense case. This is famous book, okay. So this is the Nuremberg trial, very famous trial, you can search, okay, Nuremberg trial, okay. So ultimately the Nuremberg Code was enacted in 1946. So there are different points. The main points of this is the voluntariness, informed consent and freedom to withdraw at any point of the time. So I may be included in the clinical trial, that may be phase 2 trial, phase 3 trial, phase 1 trial, whatever may be, but at any point I can exclude myself, that I do not want to be part of this. So this is any point of time free to withdraw. So this is the Nuremberg Code. Still now, this is the main tenet of the clinical trial, that is voluntary and informed consent. Anticipate scientific beneficial, that is previously we have to do some preclinical studies, you have to do some other studies, statistical analysis to see whether this is medically beneficial or not. So previously Nazi doctors do anything hypothetically. So at the first I have told about the Kurt Gödel, famous logician, that if there is some priori truth, if there is any hint of truth, then we will argue this, then we will investigate this, then we should hypothesize it. But if there is totally empirical, totally hypothetical, we should not proceed this. Okay. So avoid mental and physical sufferings. Benefits should outweigh risks. So particularly we should have a concept that there is more beneficial aspect to this rather than the risks. No intentional death or disability should be done. Protection from the harm should be always there. Subject is free to withdraw. Qualified investigators should be there. And investigators will stop if any harm develops. So this is the Nuremberg Code. Long back in the 1946 it was enacted. Then again comes the thalidomide disaster. You know about the thalidomide term. Again the thalidomide, which was mainly used in case of the pregnancy, mainly to decrease the nausea. Okay. So between 1950s and 1960s, it has been seen that the thousands of the children were born with congenital defects. This is known as the phocomalia. That is, their limbs are not grown. Okay. Mainly it happens in the Europe, Canada and the Latin America due to consumption of this thalidomide. Thalidomide is very commonly used to decrease the nausea during the pregnant times. So pre-marketing test in 300 individuals did not alert individuals to toxic effect. So ultimately there is not such efficacy studies were done. So consumers consent ultimately done, and in the United States they done some studies around the world, and ultimately they are convinced that there may be some problems with this drug. That's why this drug never entered within the United States, and some amendment, this is known as Caffey-Harris amendment, was done in the 1962. So this thalidomide was marketed in 1957 in the Germany and later in the 46 countries. This is the company Chemi Guenthal. Ultimately it alleviates the morning sickness, already you know. Dr. McBride is the Australian obstetrician who first associated with the birth defects. That is, there may be some relation between this thalidomide use as well as the phocomalia. So that in 1962 the drug was banned in most countries. Thalidomide disaster. So President Kennedy in United States praised the FDA inspector Francis Kelsey. Francis Kelsey was the FDA inspector who also hypothesized that there may be some relations to this. That's why it was not getting any marketing permission in the United States. That was President Kennedy praised, and there will be no phocomalia case in the United States of America. So thalidomide tragedy and the Kelsey's wide refusal to approve drug brought changes in the FDA. So ultimately FDA, the Federal Drug Administration, gives rise to the changes in the drug regulation that is also widespread in all of the different countries. So she was held as a drug detective, this Frances Kelsey, because of her enthusiastic inspection into this thalidomide once. The drug was not getting approved in the United States and no children were harmed in the United States. That's why he was held as drug detective, Francis of Kelsey. These amendments, some amendments came. Amendments means more stringent drug regulations are getting forced. Okay. Then came the Declaration of the Helsinki in 1964, where the different ethical principles are enacted. This is done in the World Medical Association. The foundation of the GCP was ultimately based upon this Helsinki Declaration, more or less similar to the Nuremberg Code, that is no harm should be there, etc., everything, voluntariness, informed consent, etc., etc. This is the Helsinki Declaration in 1964, where from different countries representatives came and ultimately this was enacted. Still now it was done in all of the clinical trials. It is written that it is in accordance with the 1964 Helsinki Declaration. Tuskegee syphilis study. It was done in the United States for a long period of time, mainly American Medical Research Project from 1932 to 1972, that examined the natural course of the untreated syphilis in African American men. So ultimately that leads to 28 deaths, 100 cases of the disability and 19 cases of the congenital syphilis. That is, in spite of the stringency, there are different types of the loopholes are there that ultimately leads to different types of the problems. So there are different types of the books, and movies are also there that remember this Tuskegee syphilis studies. This is also very famous. So these are the different books you can see. So only in the 1997, Bill Clinton, United States President, have said that what was done cannot be undone, but we can end the silence. That is, we need more stringent guidelines. We can stop turning our heads away. We can look at you in the eye and finally say on behalf of the American people, what the United States government did was shameful and I am sorry. So ultimately, they gives to all the family members of this Tuskegee syphilis studies approximately $9 billion compensation in 1997 itself. Okay. So it is a very important thing in case of the United States history. There are different other milestones like Henry K. Beecher, who is a famous pharmacologist, who gives a famous article, Ethics and Clinical Research. That is, need of the ethics, needs of the ethical principles in clinical research was stressed upon. He said that medicine is sound and the most progress is soundly attained. However, if unethical research is not prohibited, it will do great harm to medicine. Unethical research, ultimately it will give to great harm to medicine. Belmont report then came. There are different Belmont report that ultimately stressed upon the principle of respect, beneficence, that is beneficial aspects should be more. Principle of the justice should be there. Okay. So then comes the WHO in 1993. This informed consent, selection of the research subjects, confidentiality of the data, compensation of the accidental injury, ethical review committee, importance of this, obligations of the sponsor, these are stressed upon by the WHO, latest in the 1993. Then comes this WHO GCP, Good Clinical Practice Guidelines in 1995 by the WHO. But there are different countries gives different types of the ethical principles for the marketing authorization of the different drugs. So it needs harmonizations. A similar type of the data, similar type of the act should be there. That's why ICH comes. ICH means International Conference on Harmonization. So it is a joint initiative by the WHO, Canada, European Pharmaceutical Association, United States, European Union and Japan. So ultimately they sits and ultimately a unified code is enacted. This is known as the ICH GCP, that is the International Conference on Harmonization Good Clinical Practice. Okay. So these are the topics. Q means quality, S safety, E efficacy, M multidisciplinary. So within this, everything about the clinical trials is given and time and again it is updated. For example, these are the quality topics. How the quality of the drug that is marketed have to be ascertained. These are the safety topics. You can see different types of the safety studies are done. Like reprotox means reproductive toxicity. Mainly this is needed to give away of the teratogenicity. That is, if the drug is given in the pregnancy, there will be problems like thalidomide, like this. Then comes the efficacy studies, different types of the efficacy studies. Within this E6, that is ICH GCP E6, that mandated the GCP and did mandated the phase 1, phase 2, phase 3 clinical trials, how it is to be undergone. Okay. So E6, within this E6, everything about the clinical trials, how it is to be done is enacted. So ICH GCP, so this is the addendum, newer and newer update and addendum is given, details is not required. You can see this is the books of the ICH GCP. This is the ICH GCP sections. These sections ultimately give what are the responsibilities of the sponsor. Sponsor means those pharmaceutical company that ultimately discovered the drugs, that ultimately gives the clinical trial. What is the responsibility of the sponsor? What is the responsibility of the investigator? That is the medical practitioner who are investigating the drug. What is the responsibility of any third party? For example, anyone, statistician, any other company who are doing the clinical trial on behalf of the sponsor, like this. How the protocol is to be enacted? Protocol means everything about the clinical trial. What is the exclusion criteria, inclusion exclusion criteria, what are the follow-ups, how the different samples are collected, etc., everything. Investigator's brochure that contains every previous data about the investigational drug. That is, that investigational drug, what are the preclinical data. So if it is the phase 3 clinical trial, what is the data from the phase 1 trial, what is the data from the phase 2 trial, etc. Other essential documents. Institutional review board and independent ethics committee. So what is the composition of the ethics committee, what are the members, and whether that grants the approval of this or not, and the principles and glossary, etc., etc. Institutional ethics committee should be there for the institution. For our Santiniketan Medical College is also there is institutional ethics committee. So any research proposal, if it's to be researched, have to first undergone the ethics permission, and this ethics permission have to be done by the institutional ethics committee. So these are the core principles of the GCP. The same thing, like it is to be done by the ethically conduct, risk benefit analysis, subjects' rights is to be maintained, pre-existing information should be there, scientifically sound protocol, that is the protocol should be sound, it should be scientifically valid. Ethics committee oversight, the ethics committee should be supervise the clinical trials if there is any unethical conduct or not. Medically qualified care should be there, trained and qualified person should be there for clinical trial. Informed consent, I have already discussed about the informed consent, very important. Efficiency of the information handling and record keeping, because this data came from the patients, these are very confidential data, this should be handled with care. Confidentiality of the subject should be maintained, because this subject is suffering from this disease, this confidentiality should be maintained. Drug accountability, whether the drug is manufactured according to the good manufacturing practice or not. Quality assurance, this should be done, regular monitoring, regular auditing, etc., etc. So this is our Central Drug Standard Control Organization website, CDSCO, which is the drug licensing authority in our country. This ultimately follows this ICH GCP guidelines to perform the different types of the clinical trials. The ethical principles of the Indian GCP are in line with the ICH GCP, more or less similar, the same thing. So ultimately, lastly, who should follow GCP? Good clinical practice. Who should follow? So all the investigators, all the doctors, all the investigators, all the medical practitioners who are doing the clinical research. So when we are doing some clinical research as part of our academics, for example, when you are doing some MD or MS, you are doing some thesis, or we are doing some dissertation, again we have to give some ethics committee permission, and we have to do some all the ICH GCP. Institutional ethics committee members, clinical research coordinators, all research personnel, including the nurses, including the group D individuals, medical monitors, auditors, data entry personnel. So everything about the clinical research, and this clinical research and clinical trial is not included only in case of the new drugs. As we have already said that in phase 4 trials, we can do different other comparative trials also. Similarly, you will do some clinical research studies or trials in case of in your MD, or as a part of your academics, you can do some studies. So all these have to be followed, this ICH GCP. Everything about the clinical trial, everything about the clinical research, we have to follow the good clinical practice guidelines. So basically it is a regulatory requirement. Ultimately that ensures study is ethical and scientifically robust. It can be easily accepted, it is validated, data is reliable, safety and efficacy of the trial subjects are protected. So this is the new drug and clinical trial rule 2019, which is the updated one in our country, which is usually followed. Ultimately that leads to faster recruitment, involved patient experience, quality of the life of the patient is to be improved, improved patient data capturing, remote monitoring of the patients are included, decentralized clinical trials are included, some non-traditional data sets like some wearables, some artificial intelligence are also included in the clinical trials, so that it will be more accessible, it can be more easily performed, and the total duration of the clinical trial, that is approximately 10 to 15 years, can be shortened. For the newer drugs, for the diseases that have not been yet cured, can be obtained easily. So to conclude, ICH GCP guidelines are used in clinical trials throughout the globe with the main aim of protecting and preserving the human rights. The ICH GCP guidelines are therefore considered the Bible of the clinical trials and have become a global law which safeguards humanity as we know it today. So this is all about the clinical trials, clinical research, different phases of the clinical research, very important for your examinations also, and the good clinical practice, why the good clinical practice GCP is needed, how it is evolved, and what is the main principles of the GCP in different types of the clinical research. So thank you all.